Alright, hello everyone, welcome to another Camber video. Today I'm gonna touch on my neuroscience roots a little bit. I'm gonna answer the question for you guys, why is it that, or how is it that neurons fire? The main difference between neurons and the rest of the cells in your body is that neurons are really good at conducting electrical signals, and that leads to you being able to move when you want, think what you want, all sorts of other stuff. But in order to see the nitty-gritty of how this all works, we need to talk about what is happening at the molecular level. So just thinking about cells, similar to all other cells in your body, basically the easiest way to describe a cell is you have a bunch of organic junk and it's surrounded by a membrane lipid bilayer. And this membrane lipid bilayer basically just protects all the stuff inside of your cell from the stuff that's outside. And so we're going to have to talk about this a little bit in order for all of the electrical signaling to make sense. So as I mentioned, this membrane lipid bilayer is going to be made up of amphiphilic molecules, or in other words, molecules that are both hydrophilic and hydrophobic. So the heads of these lipids are going to be hydrophilic, and they're going to like interacting with water and charged molecules. And these heads are going to be on the outsides of the lipid bilayer. And then the insides of this lipid bilayer are going to be made up of hydrophobic, or basically neutral hydrocarbons, which do not like interacting with charged or polar molecules. This lipid bilayer allows for non-polar stuff to pass through. So for example, oxygen and carbon dioxide have a pretty easy time passing through a membrane. However, even water, which has a little bit of polarity to it, since it's small enough, it doesn't really have that much trouble getting through the basically bulk neutral inside of this membrane bilayer. Same thing with ethanol. However, once you start getting bigger molecules or stuff that is charged, such as like sugars or some amino acids, you're going to have to start using molecular transport to get those things across the membrane. And then finally, what's going to be really important for us today is thinking about different ions which are present inside cells. So the key ions that are going to be important for us to think about today are sodium, potassium, calcium, and chloride. So your cell is going to have some basic amount of these ions inside of it, and they're going to perform some given function. So just thinking about how they naturally occur inside cells, looking at this image, we see that the most amount of positively charged ion that you have inside your cell is potassium. And there's some tinier amount of potassium outside the cell. So basically the concentration gradient of potassium, since there's a lot of it inside the cell and not a lot outside, that means that it's going to want to go from high concentration to low concentration to reach an equilibrium, because in nature it really wants everything to be homogeneous and equal. So since there's that disparity of lots of potassium inside the cell, it's really going to want to come out. And the opposite is true for sodium and chloride, which are at very high concentrations outside the cell. So both of those would really love to come inside the cell. So the majority of the charge inside of a cell is going to be made up of potassium and a little bit of the chloride and sodium. And then you also have a bunch of organic anions, which is basically just a random grab bag of different types of molecules that are inside your cell. So for example, proteins such as enzymes or individual amino acids, some of those are negatively charged. Or you also have ATP or nucleotides, which make up DNA and RNA, all carry negative charge to them. All of these charges put together result in the resting membrane potential of a cell being roughly negative 70 millivolts. And this potential or voltage is basically just saying if you were to have some charge outside of the cell, how would it be electrostatically influenced by the contents of the inside of the cell? So since the inside of the cell, generally at a resting like non-active state, since it's negative, that means that stuff that is positive on the outside would really love to come in. And even though chloride is negatively charged, that you'd think that it wouldn't want to come inside the cell, but since there's so much more of it outside of the cell than inside, it would still want to come into the cell to some extent because there's such higher concentration of chloride outside and it wants to even it out. So even chloride being negative would want to come into the cell if it could. Now looking at a better image, looking at, I guess, the inside of what a regular cell would look like. And this happens not with just neurons, but all cells in your body keep a negative voltage or negative membrane potential, although neurons generally keep it at negative 70 millivolts. So looking at the composition of the ions here, what we can see is you have a bunch of these potassium ions and very few sodium ions on the inside of the cell, which is shown on the bottom there. And you might be thinking, since it is that nature loves an equilibrium, it loves that everything is equal and equally distributed, how is it that you get so much potassium inside the cell and so much sodium outside the cell? And that's where we get to sodium-potassium pumps. And this pump is very important, plays a key role in your body. And what it does is, using ATP, or the energy currency of your cell, you're taking 3 sodium ions from inside your cell and exchanging it for 2 potassium ions from outside the cell. And that is how you get that distinct high amount of potassium inside and high amount of sodium outside. And since you're taking sodium from the inside where it wants to be to the outside where it doesn't want to be, because there's already a lot of sodium out there, you need to use some energy to work against the concentration gradient to get this to work. Another interesting fact about this sodium-potassium pump is that in most of the cells in your body, this takes up roughly 20 to 40% of all energy usage is running this sodium-potassium pump so that your membrane voltage stays roughly the same in all cells in your body. However, neurons in your brain, since basically their entire function is to conduct this electrical charge, almost 70% of the energy in your brain goes to powering this one pump, which I think is kind of insane. But now that we know basically the contents within an average neuron, you have a high amount of potassium inside and a high amount of sodium outside. Let's look at the structure of a neuron itself. So the real way that neurons differ from regular cells is that they have 2 key, I guess, structures which differentiate them. That is that they have axons as well as dendrites. So the cell body, this is going to be very similar to other types of cells that you have, very similar in composition, very similar functions. However, then you have axons, which these axons, what they do is they're going to transmit this electrical signal down to the axon terminals. And then at the axon terminals, the goal of these axon terminals is to transmit the electrical signal to dendrites. So axon terminals are going to synapse, or basically form connections to not hundreds, thousands, if not tens of thousands of dendrites of other neurons. And there's this high amount of interconnectedness from axon terminals to dendrites. Based on the, I guess, charge being the electrical signal being taken from the axon terminal to the dendrite will then influence whether that downstream neuron is going to fire itself or not. So looking at what happens when a neuron fires or produces an action potential, as it's so called, what we're seeing here is that the resting membrane potential, as I said, you're going to have a lot of potassium inside the cell and a lot of sodium outside. Then you have this initial period where you have some sodium coming into the cell, and it's going to increase or make the membrane potential less negative. What this then causes is it makes voltage gated channels, sodium channels at the axon hillock, then open up and then cause more sodium to rush into the cell. Since you have a lot of sodium rushing into the cell and you're keeping the same amount of potassium in there, you're going to have suddenly a lot more positive charges inside the cell than you normally would, and this causes the membrane potential to become more positive. However, you want to eventually get back to this negative resting membrane potential. So, and as you remember, since there's a lot less potassium outside the cell, if you open up a potassium channel, you can then get rid of some of these positive charges by then getting rid of that potassium, and then you can start getting a lower membrane potential. Then once you have some of that potassium leaking out, then you can turn on sodium pumps and get rid of all the sodium that then came into the cell, and then you get back to where you started at resting membrane potential. So talking about these voltage-gated sodium channels, they're present at the axon hillock, and at the axon hillock, that is where the action potential starts, or where this electrical signal basically starts down the axon towards the axon terminal. These voltage-gated sodium channels, they don't respond to any ligand or neurotransmitter, and instead they only open based on changes in voltage in the surrounding environment. So if you have more sodium ions nearby these voltage-gated channels, it'll cause a change in the conformation of this ion channel, and it'll open up a channel which allows sodium to pass into the cell. And there's a selectivity filter, so to say, where ion channels are specific for only a specific ion. So even though sodium and potassium are very similar in size, these channels, through many millions, if not probably billion years of evolution, have become very specific for that type of ion. So you're only going to have sodium come flowing in. But anyway, once you reach a high enough membrane potential, then these channels will open and allow sodium to flow from the axon hillock into the axon, and then you're going to start to have this electrical signal go down the axon until it reaches the axon terminals. And looking at this GIF here, what we can see, there's basically this propagation of positive charges going down the axon towards the axon terminals. And you might be wondering, because it makes sense that you have the electrostatic interaction of the positively charged sodium wants to go into the negatively charged cell, but you also have that concentration gradient keeping the sodium from, or letting the sodium come into the cell. So then the question becomes, why is it that you only have this electrical signal going one way from the axon to the axon terminals? And that is something that I neglected to mention when I was talking about these voltage gated channels, is that they open for only a specific amount of time and let only a specific amount of sodium ions into the cell, and then they enter what is a so-called refractory period, where then you have these channels get plugged up and basically aren't allowed to open again, and they basically shut down for a short period of time. And while these channels are in the refractory period, you can have potassium channels open up, which decrease the membrane voltage. And then you can also have sodium potassium pumps pump out the sodium, so you reach resting membrane potential, and by the time these channels are out of their refractory period, there's already not enough change in voltage for these channels to open again, which means that this electrical signal will essentially only travel in one direction. Although this is not fully true, and there are cases when this does happen, but for the sake of this video, this electrical signal only travels from the cell body to the axon terminals. Since there's some neurons in your body that are pretty long. For example, if you think about the neurons which control the movement of your foot, you're going to have the signal coming from your brain, and it has to make it all the way down to your foot. So the axons of these neurons will be almost up to 1 meter in length. And as I mentioned, the sodium-potassium pump takes up more than half of the energy that your brain uses up. And so if you needed to keep this electrical signal going for a full meter, you'd be taking a lot of energy to keep this signal happening. And so your body has a pretty ingenious solution as to how to make some axons more efficient at transmitting this electrical signal. That is through the use of Schwann cells or oligodendrocytes. Schwann cells are basically these types of cells in your peripheral nervous system. Oligodendrocytes are the counterparts which are present in your brain. And Schwann cells essentially are just cells which wrap themselves around axons and produce fatty myelin sheaths, which are very similar to membranes in which ions can readily pass through these myelin sheaths. And so what this makes is that you won't have a loss of, or you won't need to conduct this signal through the whole length of that axon, because a majority of it is going to be covered by Schwann cells and prevent the ions from leaving the axon. But you can have the full axon be covered because there's only so much concentration gradient driving the sodium from going towards the axon terminal. And what I mean by this is if you have only a small amount of sodium and you want it to travel a full meter in length, you're going to need a lot of sodium for it to continue wanting to expand and go out towards that way where there isn't any sodium at the very end of the axon terminal. So you're going to have some places where you can reintroduce some sodium to keep that electric signal going down the neuron. So you have nodes of Ranvier, which are basically gaps in between these myelinated sections, which allow for more sodium atoms to come into the neuron and continue propagating that electrical signal down to the axon terminals. And so what happens now when you get to the axon terminal? So you're going to have a bunch of these sodium ions coming in, and this changes the voltage of that axon terminal. What this causes then is for neurotransmitter to be released and then land onto some ion channel which is present on the dendrites of some secondary neuron. So how does this happen? The change in voltage in the axon terminal causes a different voltage gated channel, this time a calcium channel, to open, and calcium will come into the axon terminal. Calcium is so high on the outside of the cell. There's almost 10,000 times more calcium on the outside of your cells than on the inside, whereas sodium is roughly only a couple hundredfold more on the outside of the cell than on the inside. So calcium really, really wants to get in. So when there's a change in voltage, this calcium will basically sprint into the cell and it will start interacting with proteins on vesicles which contain neurotransmitter. So these vesicles are going to contain molecules called neurotransmitter, or basically types of molecules which can bind to receptors on dendrites and continue the ion flow into dendrites and continue propagating that electrical signal. So looking at the structure of this membrane vesicle and how it actually dumps its neurotransmitter into the synaptic cleft and towards the dendrite. This vesicle has a bunch of proteins attached to it. One of them, synaptotagmin, is a protein which, in the presence of calcium, causes a shift in its structure, and it makes this vesicle, which is nearby the membrane of the axon terminal, become even closer. And since the vesicle is made up of similar amphipathic molecules, the membrane of the vesicle would like to interact with the membrane of the axon terminal membrane. And in the presence of calcium, you're going to have basically a pulling of these snare proteins, which are going to pull the vesicle closer and closer to the membrane until they basically become one, and the inside of the vesicle becomes the outside of the cell, and this neurotransmitter can now be free to float around and bind to receptors on dendrites. These neurotransmitters, they have time to be able to bind to these receptors, but then eventually they either get taken up back into the axon terminal, degraded, or they just simply float away and can no longer interact with that specific dendrite that they're supposed to bind to. So these neurotransmitters are now going to bind ligand-gated ion channels. So these ion channels aren't affected by voltage at all, but if a specific ligand or neurotransmitter binds to the channel itself, it will cause the channel to open, and similar to a voltage-gated channel, will allow some ion to pass into the dendrite. So in the case of an excitatory channel, you're going to have sodium come into the dendrite, or an inhibitory channel, which is going to allow chloride to come into the channel. Now, putting this all together, in the cell body, if the resting membrane potential gets above some threshold level, which is generally negative 55 millivolts, then the voltage-gated channels in the axon hillock cause sodium ions to flow into the axon, and the signal is going to be propagated towards the axon terminal. These sodium ions will then cause voltage-gated calcium ion channels to open, and then calcium will cause vesicles with neurotransmitters to fuse with the membrane of the axon terminal. And these neurotransmitters can then bind ligand-gated ion channels on dendrites, and then this will cause an influx of either sodium or chloride into the dendrite. And these dendrites are going to interact with many thousands, tens of thousands, maybe even hundreds of thousands of axon terminals from other neurons. And so the summation of all the ions which are coming into the dendrites are going to meet up in the cell body. So imagine that you have some neuron and half of its dendrites are getting an excitatory response, or basically half of the dendrites are letting sodium in and the other half are letting chloride in. Since you're getting equal parts of each, you probably won't have a change in the membrane potential of that neuron. And once they reach the cell body, there won't be any change, and it's still going to be at resting membrane potential, meaning that the voltage-gated sodium channels in the axon hillock won't open up and the signal is going to die there. However, if you have a lot of these ligand-gated ion channels letting in sodium, if you sufficiently depolarize or make the voltage higher in the cell body, then you can continue propagating that electrical signal. And through very intricate circuits, that leads to us being able to see, hear, and do anything and everything that we have ever known and loved. I hope that this was a pretty cool look into neuroscience. This is, I think, just one of many videos that I'll do going into the field of neuroscience. If you thought this video was exciting and or wanting to know more, feel free to hit the subscribe button, maybe leave a like if you feel like it. And as always, I hope you guys have a good one.